Hello students. In previous lecture we have studied the types of growth and growth rate and all the introductory part to this chapter, that is plant growth and development. In this next part we are mainly focusing in this lecture about development. Okay, so development, if we use this term, what it means first that we should understand. So it include all changes that an organism goes through during life cycle, from germination of seed to senescence. So this changes from seed germination to the particularly senescence is known as what? Development. So from seed, what are the sequence? Then initially we have a seed, then seed germination will occur. After seed germination they will have a juvenile phase or vegetative phase. Then after juvenile phase they particularly carry out reproductive stage, and in reproductive stage they develop a flower. So flowering will occur. After flowering, fruiting will be observed, which is a part of flowering, and fruiting is part of a reproductive stage. After fruiting, again seed will be obtained, and this seed will start a new generation. So this is the sequence how the development proceeds. But when we talk about, this is the development in case of the plant, whole plant, a development stages of the whole plant. But when we talk about the developmental stages of a cell, developmental process or stages of plant cell. So when we talk about developmental stages of plant cell, we know that initially we have meristematic or dividing cell. Now meristematic cell or dividing cell first grow in its protoplasm, which is known as plasmatic growth. After growing, when the plasmatic growth or protoplasmic growth will occur, what will happen? The size of the cell will increase, expansion will occur, and nucleocytoplasmic ratio will be disturbed. So now after plasmatic growth, cell have a two pathway. Either is go on elongation, then differentiation, then maturation, and ultimately we get a mature cell. Maturation, and we will obtain a mature cell, and this mature will undergo senescence and death will occur. So that is the one way in which the cell will expand, differentiate, mature, and mature. After maturity it will die. But another way after this is either it keeps on dividing. So if again cell division will occur, then meristematic cell will be obtained again. So this is the way how cell proceeds through some of the developmental stages. So these are the developmental stages of a plant and particular cell. So when plants show developmental stages, their cell are also showing this type of changes, and due to which the plant follow different pathway. So when plant follow different pathway, this phenomena is known as plasticity. It is known as plasticity, and plasticity defined as when plant follow different pathway. Plant follow different pathway in response to environment or in response to environment or phases of life to form different kind of structure, to form different kind of structure. This ability is known as plasticity. So plasticity is what? Ability of a plant to follow different pathway in response to the environmental phases of life. And due to following a different pathway, when it forms a different structure, it is known as plasticity. And the example of plasticity is heterophyly. Now heterophyly means that is a difference in shape of leaves. Heterophyly means difference in shape of leaves of plant. Now different in shape of leaves due to the different environmental factor. So it is an example of heterophyly, and this heterophyly is of two types. One is environmental heterophyly. And another one is the developmental heterophyly. Now when we are talking environmental heterophyly, then it means the shape of leaves is different in water and terrestrial habitat. Means the plant is same, but it has a different shape of leaves in water and terrestrial habitat. So if the plant has a difference in water and terrestrial habitat, then the shape changes, and the example is buttercup. Buttercup is also known as Ranunculus flagellaris, a scientific name of this. But another type of heterophyly is a developmental heterophyly. In this, shape of leaves is different in juvenile phase and adult phase. So in adult and juvenile phase, the shape of leaves is different, then it is known as developmental heterophyly, and the example are cotton, coriander, and larkspur. Cotton, coriander, and larkspur. So this is about heterophyly and plasticity. Now we know that growth, differentiation, and development. Growth, differentiation, and development are very closely related event. Very closely related events in the life cycle of plant. And we know that broadly development is considered as a sum of growth plus differentiation. And when we are talking this development is under the control of intrinsic and extrinsic factor. So development is influenced or under control of extrinsic and intrinsic factor. Now when we are talking about intrinsic factor, this intrinsic factor are also of two type, that is intracellular and intercellular. Intracellular and intercellular. Now when we are talking about intracellular, means genetic makeup of a cell regulating all the activities of a cell, and it is present within the cell. Because of this we call as a intracellular intrinsic factor. But when we are talking about intercellular factor, it is a chemicals such as plant growth regulators, such as plant growth regulators, that are PGRs. Okay. Now apart from this, that is the intercellular component and intracellular component, there is an extrinsic factor. And when we talk about the extrinsic factor, this include light, temperature, water, oxygen, nutrition, etc. Okay, so all this become the extrinsic factor. Now out of this three intercellular extrinsic factor, for our purpose and with respect to this chapter, plant growth regulators are important. So in the next class we are going to discuss mainly the different plant growth regulators. Now this plant growth regulators, that is PGRs, are of two type: plant growth promoters and plant growth inhibitors. Those plant hormone or chemicals which help in a particularly growth promoting activities of plant, they are known as plant growth promoters, whereas which involve in inhibition activities or negative activities of the plant, they are known as plant growth inhibitors. Now plant growth promoters include auxin, gibberellin or gibberellic acid, and cytokinin, whereas plant growth inhibitor include ethylene and ABA, that is abscisic acid. Now in this, ethylene has growth promoting activities as well. Important point is ethylene has growth promoting activities as well. So it is growth promoter as well as inhibitor. So it is a growth promoter as well as growth inhibitor. So out of all this five, we are going to discuss one by one. First auxin we will discuss, then gibberellin, cytokinin, then ethylene, and then abscisic acid. We will discuss one by one all this particular hormone. And in this, first we are starting with the auxin. So we start with the first plant growth promoter, that is auxin. Now auxin, if we talk about, the precursor of auxin is tryptophan, that is an amino acid. It is an amino acid which is a precursor. Now, after this, if we talk about the first type of auxin or the particular chemical nature of an auxin. Okay, so auxins are weak organic acids. They are nothing but weak organic acids, and this auxin are classified into two groups, that is natural auxin and synthetic auxin. Two groups, that is natural auxin and synthetic auxin. The natural auxin occur in plant naturally. They occur in plant naturally, whereas when we talk about synthetic auxin, they have a similar actions and they are synthesized artificially or by human activities. When we talk about this natural auxin, first natural auxin was isolated by Kogel and Hagen-Smith. Kogel and Hagen-Smith in 1931 from human urine samples of a person suffering from pellagra, that is a deficiency of niacin or niacinic acid, and he was identified it as auxin A, auxin B and heteroauxin. And in this heteroauxin, which is also known as real auxin, that is indole acetic acid. It is known as indole acetic acid. So indole 3 acetic acid is a natural auxin. Besides this, indole butyric acid is also a natural auxin. Indole butyric acid is also a natural auxin. So these are the two natural auxin which was found in a plant. Okay, natural. But next is the synthetic auxins. So when we come to the synthetic auxin, they are synthesized artificially and they have similar activities to the auxin. Some of these are NAA, that is naphthalene acetic acid, then 2,4-D, that is 2,4-dichlorophenoxy acetic acid, and 2,4,5-T, that is 2,4,5-trichlorophenoxy acetic acid. And NaAM, that is naphthalene acetamide. Apart from this also there are several synthetic auxins, but these are the one of the known synthetic auxins which you should remember. Okay, so this is the natural and artificial auxins. Now after natural artificial auxins, next we talk about the transport of auxin or a distribution of auxin. So next is transport or distribution of auxin. Now actually auxin is synthesized in the shoot and root apices. Auxin synthesized in root and shoot apices, and these are produced in relatively small amount in the root. Produced in very small amounts in root. When we talk about the particular shoot and root phenomena, we come to know that in shoot it gets synthesized at the apex and transport occur towards the base. Okay, so it is transported primarily towards the root through the parenchyma. So this transport is known as basipetal transport. But when it moves from the base of the shoot to the root apex, here towards the root apex, so it can be considered as acropetal transport. But overall it is going towards the base, so the transport is basipetal and polar. Basipetal and polar transport, it is known. Now with respect to auxin, it is known that in auxin, low concentration of auxin stimulate growth. It stimulates growth, while high concentration would inhibit the growth of same cell or organ. Now it is known that root in general show growth initiation at very low concentration, that is 0.0001 parts per million, while in stem the similar response occur at relatively high concentration, that is 10 parts per million, show similar response. So these are the particularly different distribution patterns and how they show their effect that we have understood. Now next we talk about the bound and free forms of the auxins. Next, auxins are of two types, that is free auxin and bound auxin. Now free auxin occur freely in the cells and they are in free state, and this is active, and if it is a free state and active, it can be easily extracted. We can extract easily, we can isolate easily. But when we talk about the bound auxin are not in free state. they are bound with the chemical, any chemical, they are inactive and they are difficult to, they not extracted easily. So they are meant for storage, not for transport. And the example are indole acetic acid, aspartic acid, indole acetic acid, inositol, indole butyric acid, alanine. So these are the different forms in which the bound auxin is found, which is used for the storage. Students, now we talk about, after studying these types of auxin and the bound free auxins, we discuss about the discovery and the distribution of different scientists in the discovery of auxin. Okay, so we know that auxin is the best not known plant growth regulator. Auxin is best known plant growth regulator. Now when we talk about the discovery, earliest experiment was done by Charles Darwin and his son Francis Darwin. Now this Charles Darwin and Francis Darwin are the scientists who worked on seedlings of canary grass, and this canary grass is scientifically known as Phalaris canarensis. So he worked on this canary grass, that is a canary, Phalaris canarensis, and what he observed that when a particular coleoptile or canary glass coleoptile given a unilateral light, it showed bending like this. So bending of coleoptile occur when there is a unilateral illumination of light on the tip of this particular coleoptile. But when this coleoptile was decapitated, means cap was removed, then it become like this and it do not show bending. So no bending occurred when there is a decapitation occurred. When this tip was covered by metal foil, then also no bending. But when the tip was covered by translucent cap, it show bending. Means the light passes, stimulus passes through this translucent cap and received by the tip, so bending occurred. So this shows that when the tip receives the light or unilateral light from the one side, then this show the tip show the bending and a bending response would occur. So it led to the Darwin to conclude that, so Darwin concluded that certain kind of influence is generated at the tip is generated at tip, which is then transmitted to base where growth takes place. So what this influence that he was not able to identify, so influence remained unidentified. After Charles Darwin and Francis Darwin, second scientist came whose name is Auxin and Jensen. Whose name is Auxin and Jensen. Auxin and Jensen observed that coleoptile with decapitated tip neither grow nor bend towards light, means they do not show phototropism. Okay, but when this tip was removed, that is, a decapitation occurred, and then after decapitation we put a intervening block of gelatin and cap was again kept here, this show bending, means through gelatin this chemical passes due to which bending occurred. But when in this coleoptile, if we keep a mica plate, then it shows no bending, means it shows that influence is chemical diffuse, is a chemical that diffuse through gelatin block. This can diffuse through the gelatin block, so it can pass through the gelatin block. That was discovered by the Auxin and Jensen. Later on, one more scientist, Paul, demonstrated that coleoptile would bend even in dark after certain treatment. It can even bend after a certain treatment. And later on, the important scientist, that is F.W. Wendt. So the contribution of F.W. Wendt is important with respect to exam. So be remember. Now what he did, he taken oat coleoptile. He worked on the oat coleoptile, and oat is Avena sativa, a scientific name. Here first decapitation occurred. So in decapitation, cap was removed. Now this cap was placed on agar block. So when we put agar block and over agar block we keep this cap. Now after certain period of time we have a decapitated oat coleoptile over which agar block was placed. When agar block was placed on the decapitated coleoptile, the bending occurred like this. So this shows that agar block has that chemical which is transporting through the gelatin, which is transporting through the tip. So that is the chemical present in agar block, means it can be transferred to the what, a agar block. Okay, so it is transferring from here to agar block, and then agar block is placed here, so it shows the bending. So what it concludes that the influence or chemical influence responsible, so Wendt showed that chemical influence responsible for this phototropic response is Auxin. And when we talk about Auxin, it is a Greek word, it means to increase or to grow. So Wendt is the scientist who credited for discovery of Auxin. So we remember this is the name of the scientist who worked on the haute coleoptile and isolated this Auxin. Now after the discovery part, next we talk about the functions of Auxin. Next we talk about the functions and applications of the Auxin. In functions of Auxin, when we study, we come to know the first function, that is cell enlargement. First function is cell enlargement. So Auxin causes cell enlargement by solubilization of carbohydrates, loosening of cell wall microfibrils, synthesis of new cell wall material. So by all this, what will happen? With the solubilization of carbohydrate will occur, then solute will increase, that lead to the absorption of water, and due to the increase in water content of the cell get enlarged. So at that same time, loosening of cell wall also occurs, the new cell wall material also gets synthesized, and that lead to the what? A particular cell enlargement. And here increase in respiration is also observed. Respiration also increases during this. After this, second is prevention of lodging. Prevention of lodging. Prevention of lodging says that lower internode of stem of cereals is weak and long. So as a result, plant bends down or droops, known as lodging. So this application of synthetic Auxin, that is naphthalene acetamide, lead to prevention of this bending or lodging. Okay, so lodging is a weakening and drooping or bending downward of the particular plant. Third is apical dominance. Now first we should understand what do you mean by apical dominance. Suppose this is the any plant and this is the apex. The time till which apex is present, the lateral bud will not grow. It does not allow lateral bud to grow. Why? Because it will allow all nutrient to reach apex, not to the lateral bud. So apical dominance is defined as apical bud inhibit the growth of lateral bud. Apical bud inhibit the growth of a lateral bud, and this condition is known as apical condition, apical dominance. Now, so this is the apical dominance. So if we remove this tip, then there is a chances that this lateral bud will start growing. Yes or no? So pruning or removal of shoot tip of tree like tea and hedge plants. So in tea and hedge plants, if pruning is done, then it helps to remove the apical bud and lateral bud will grow. At result dense growth of hedges is observed. Dense growth of hedges is observed. Hedges are nothing but a bushy plants. Next, fourth function of auxin is responsible for phototropism and geotropism. Responsible for phototropism and geotropism. Fifth, promote root initiation. Promote root initiation and root initiation in callus and stem cutting. In a callus and stem cutting. In a stem cutting, if root initiation is induced, then it is induced by indole butyric acid and naphthalene acid are used to induce root initiation on the stem cuttings. Sixth, it delay or prevent abscission. Abscission is the falling of leaves, flower and fruit. It delay abscission in younger plant parts and promote abscission in older plant part. Now abscission means falling of leaves, flower and fruit, and this is the one of the important point that you should remember. It is an NCERT point and it is important. Next, it induces parthenocarpy. It induces parthenocarpy, and parthenocarpy is production of seedless fruits. It is a production of seedless fruits. Example is tomato. Next, after that, it increase number of female flower in plants like cannabis. Means it is responsible for femaleness. Auxin is responsible for the femaleness. Next, seasonal activities of cambium promoted by auxin. Then it promote xylem differentiation. It promote xylem differentiation, and it also responsible for healing of injury through auxin induced division of cells in an injured area. Next, it also used as vidicides. It is also used as a vidicides. In this, synthetic auxin like 2,4-D and 2,4,5-T, they used to remove broad-leaved weeds or dicots weeds in monocot fields. So wherever the monocot plant is growing, in which by chance if dicot or broadleaf weeds will grow, that will be removed by the vidicides. 13. It promotes flowering, that is like naphthalene acetic acid and 2,4-D promote flowering in lychee and pineapple. 14. (2,4-D) for citrus fruits and NAA for tomato. They cause pre-harvest fruit drop. Before harvesting, the fruit drop will be observed when there is a 2,4-D is used for the citrus fruit and NAA for the particularly tomato. So these are some of the functions and applications of the auxins. Now when we talk about the antioxidants, antioxidants inhibit activity of auxin, and they are: first is TIBA, that is triiodobenzoic acid, and PCIB, that is parachlorophenoxybutyric acid. So these two are the inhibit activity of auxin, so they are antioxidants. So they actually inhibit the transport of auxin to the lateral buds, promote the transport of auxin to the lateral bud by the presence of this. Next we talk about the last part, that is bioassay of auxin. Bioassay of auxin will determine the concentration at which the particular physiological effect will be observed. So the bioassay which are Avena curvature test. Now be remember, Avena curvature test is important because it was asked twice in NEET. One time they have asked that Avena curvature test is a bioassay of. So in option they have given auxin, gibberellin, cytokinin, so answer becomes auxin. But in one after that in next year they have asked that the bioassay of auxin is. So you should remember this is the easy question. Second is split P test. And third is the root growth inhibition test. At high concentration root growth will be inhibited. So these are some of the bioassay of auxin. So till now we have discussed with the all part of all component, all functions of the auxins. Next, after this we will start with the another plant growth hormone, that is gibberellin.